Thank you, Athena, and thank you, Carlo and Joe, for organizing this super fun event. I had a lot of fun making these slides, so I hope you guys have a lot of fun listening to them. And before I started putting my talk together, I should have researched it a little more because there is such a thing as a mom bee. And so that should I want to readjust the title of my talk to include the mom bee in it. Okay, and I just wanted to briefly acknowledge the people and individuals that were a part of this project from the beginning. Athena, Melissa, and Angelo are all here at ASU. And then so a lot of the design work in the slides and the t-shirts were from Mike Boddy. Okay, so I wanted to start off with a few definitions. The first very important definition is what is a mom bee. A mom bee is, well, it's a noun. It resembles an actual person characterized by a staggering walk, a blank stare, caused by having one or more children, and the known cure is sleep, but also treated with coffee. And I think a lot of people, it was like I planned that or something. And I think a lot of people would really agree that this is a really nice definition of how you feel after having children. Okay. The second definition I wanted to give, which was in the title of my talk, is microchimerism. A lot of you might not be familiar with this term, and when I say the term microchimerism, I mean a small number of cells from a genetically different individual, okay? Microchimerism occurs naturally. These transferring from cells can happen in two individuals in multiple different ways, okay? One of those ways is in organ transplantation, okay? You do get a small number of cells or an entire organ from another individual, and you coexist. Another natural way of obtaining microchimerism is through twinning. So twins in utero can swap cells. And then last, and what I'm going to focus on for this talk, you can get microchimerism from pregnancy, okay? There's a swapping of cells between the fetus and the mother. It's a bidirectional transfer of cells during pregnancy, okay? And this happens to everyone, okay? Where everyone has been born and thus has been a recipient of someone else's cells, okay? Okay. So after going through the definitions, I kind of wanted to give you a case study. This is a really interesting case where an individual, Lydia Fairchild, she went into a custody battle over her children. She was getting divorced from her husband. They did paternity testing, but they also did testing on her as well. And what they found was that she was not genetically the mother of her children, okay? And this confused the courts, and they thought clearly there's something wrong. They thought she was lying or deceiving the court. So she was also pregnant with her third child when she was going through this custody battle. And so the court had an observer present and in the room with her, watched her give birth to the child. That's kind of weird. And then they genetically tested that child and the baby. And again, the DNA test suggested that she was not the mother of the child. We'll go back to that in a minute, but when you observe something and then the results don't match up, you might think there could be something wrong with your test or something else is going on. But indeed, the court was not pleased with this outcome. So how does this happen? And I wanted to back up a little bit and discuss the biology of pregnancy because I think it's really important for setting the framework of studying microchimerism. So way back to us being placental mammals, right? So unlike our egg-laying cousins, we're mammals. We have internal gestation, okay? And this transition to internal gestation, which happened over 100 million years ago, the mother took on a lot of risk, some cost and benefits from growing the fetus within her body, okay? But one of the most and I liked a lot that this talk is following up with Lee's talk, because we talked a lot about risks. So it's like absorbing some of the risks, but then it's risky to now have something growing inside you that has direct access to your resources and that can manipulate you, right? And so in placental mammals, the placenta is an endocrine organ that attaches to the maternal wall in her body and transfers resources from the mother to the baby. Okay? That's mind-blowing. It's super amazing. The fetus creates an organ that attaches to the mother and extracts resources from it. Okay? Yes, you're still getting resources. And how we all know, and it fits nicely with what Lee and Athena were talking about today, is that resource transfers can be very cooperative, right? Think about, in terms of zombies, maybe if there's enough brains around, you're sharing brains with each other, right? But these resource transfers, especially when they're limiting resources, you have different genetic interests, it can lead to conflict over these resources and who gets how much of what, okay? Especially when there's two separate genetic entities trying to get the same resources, okay? Remember, the fetus is 100% related to itself, but it's only half related to the mother. The mother's also only half related to the fetus because there's paternal genes going on there, okay? And this placenta actually hijacks the maternal resources, okay? So this leads me to my first point in Momby Lessons 101, that babies are needy, all right? And this is a theme. This is what we really need to, if we want to create a Momby, what we really need to know is to first establish is that we know that babies are extremely needy, especially mammals, okay? Okay. And how does this really play out? This is something in the literature called maternal-fetal conflict, and those of you that are not familiar with it, there's a lot of great work showing that the fetal's interests are to extract more resources. It's 100% related to itself. And it's in the mother's interest to limit these resource transfers. This is not maybe the only offspring she's going to have. She needs to save resources and divide them equally across all of her offspring to give them a fighting chance for survival, right? And you see this tug of war over resources in placentation. And it's this optima of the fetus wants more than the mother can give, and the mother's pushing back. And when everything is equal, you get this happy, healthy pregnancy, right? What's really amazing is you can see this maternal-fetal conflict playing out in the evolution of the placenta. And it's one of the coolest things in, I think, evolutionary biology is how different the placenta is across placental mammals, right? It's the most diverse mammalian organ. So these aren't real pictures. I think they're like 3D reanimated of ultrasounds. But what you really see here is you have this, and I know it's really hard, but you have this diversity in shape. So sometimes the placenta can be discoid in shape. Sometimes it's like a band around the individual or the organism. And sometimes it's little tiny dots diffused throughout the mother's endometrium. And I thought it would be relevant to compare brains and hearts because it's a zombie conference, and you don't see this diversity in other organs. You don't see this like intense morphological diversity in other organs. And we think this placental diversity is because there is an evolutionary consequence between the mother and the fetus. And so these are categorized in, like I said, the shape of the placenta, but you can also categorize the placenta in its degree of invasiveness within the mother, okay? And so you have going from, what side are we at in here? The left to the right, you go from least to most invasive. The fetal tissue in epithelial chorial placentas only breaks through the first layer of the maternal wall, endothelial breaks through the second layer, and hemochorial breaks through all three layers of the maternal and endometrium tissue and actually hooks on to the maternal spiral arteries, remodels the mother's arteries to get direct access to resources, okay? And not surprisingly, humans have the most invasive placenta of all mammals, okay? We've actually built up extra tissue. Over evolutionary time, we think this is one reason why women, humans, menstruate, is because we build up all this protective layers of tissue. And we differentiate this tissue so that potentially the trophoblast cells, or the specialized fetal cells, don't invade too far, all right? So this has been this ticking up of this evolutionary tug-of-war between the fetus and the mother, the fetus wanting more resources, the mother blocking it, the fetus blocking those blocks, and then the mother blocking those blocks. And we can see that play out at the molecular level, which I won't go into detail right now, but I'm hoping to leave a lot of time for questions. Okay, so now that you have Placenta Biology 101, let's go back to microchimerism and why potentially you could make a mombi. So the first rule, so I said babies are needy, but the first rule you need to know about a mombi is babies want your resources in the womb, okay? And we've seen this through this evolutionary tug-of-war between each other, okay? And it's important to note that this has caused pathology in mothers, okay? If the fetus tries to extract too much resource in a condition called preeclampsia, where the mother's body is actually ramping up her blood pressure, sometimes it's the fetus demanding more resources. And it can be lethal in the mother if she doesn't get to the hospital, okay? And so this tug-of-war can really railroad and cause health consequences in the mother. Okay, so let's go back to our case study of Lydia Fairchild. We learned that they observed her, she had children. Her DNA test did not match her children. And so what they ended up doing smartly, I think they literally almost took custody away from her. They finally did a cervical sample, and that DNA matched her children, all right? And so, whew, okay, so she was indeed, even though she gave birth to it and they observed it, they still followed the DNA evidence. She was indeed the mother of the children. She had DNA proof now, and she was allowed to have custody of her children, okay? And how did that happen? Doctors think, but not sure, that she actually had twin cells. And she was a chimerism of both her twin and her own cells. She was carrying two different sets of DNA. Although it couldn't be empirically tested, it seems like this could be a logical explanation for it. But that starts getting into the unknown. And really, an important question to ask is, how can cells from two different humans exist in one body, all right? So when I was taking all the biology courses that I took, in grad and undergrad, they tell you in Biology 101, your immune system learns the difference between self and non-self, okay? And anything that is non-self gets trained and the immune system can clear it, okay? And so what we're seeing here is this biological phenomena, and you can have a woman who had a healthy life and children, but she had two different sets of genetic material in her from two different individuals. And how is this really possible? But what we're finding out is it's a normal biological process. We are all chimeras, all right? We have all had that transfer of cells in utero. Some people are at more extreme levels of it than others, but we really all are chimeric, okay? And it's really because of this super invasive placenta, the fetus trying to extract more resources, now directly accessing the blood flow. It can slough off cells, but the cells from the mother can now transfer into the fetus as well, okay? So there's this swapping of both the fetal and the maternal DNA, okay? And so what do we know about this? What do we know about these biological processes now that we know it exists? We know that, I mean, it's been quite a few years to see how common it is among women. It seems very common. Where's the current status of what we know about microchimerism today? Okay. So during pregnancy, the fetal trophoblasts invade the maternal tissues, and you start seeing fetal cells frequency increase in the maternal blood with increasing gestational age, okay? It starts around 7 weeks of implantation. The tricky thing here is that some women do not know they're pregnant, and we know that there's a high rate of miscarriage, and you will never know that there was a sloughing off of cells into the maternal body, okay? We know that fetal cells have been detected decades after a mother has given birth, so they linger, they stay along for a long time. They happen in every pregnancy, and we detect them decades after a mother has given birth to her offspring. And the really cool thing is we're starting to find it in every tissue in the maternal body. Every tissue they look at, they're finding fetal cells. Not in all women and not in all frequencies, but they do find fetal cells in almost every tissue of the maternal body. Typically, so the technology right now is a little tricky. So typically, it's very hard to distinguish one cell from another, especially when 50% of the DNA is identical to the host that you're looking at. So for microchimerism studies, they're usually looking for Y chromosome, right? And so it's a subset of women that they're looking at, so women who have given birth to a son. And then they look for Y chromosome integrated in these tissues, okay? And so I would argue that there might even be an underestimation of how much we're seeing microchimeric cells being transferred between the mother and the fetus. Okay, what do we know about what tissues they hit and how often they're in it? It seems to be pretty standard in both mice and humans that the highest frequency of microchimeric cells are in the lung. And then it goes to spleen, liver, kidney, brain. There's microchimerism in the brain. I'll get back to that in a minute. But the patterns hold for both mouse and humans. There's not a lot of studies looking at across mammals in microchimerism in general. Interestingly, mice also have a hemocorial placenta, that really invasive placenta type that breaks through and remodels the maternal arteries. Not all mammals have that placenta type. Cows and dogs do not have that placenta type. We still detect microchimerism in cows and dogs as well. Other than that, it just has been very understudied. So we assume it's across all placental mammals. We would also assume that the frequency might be higher in organisms that have a hemocorial placenta type, just because there's a direct transfer of the cells, but we are seeing it across all placental mammals. So during pregnancy, this happens. How is this possible? One, how is it possible that there's an organ that attaches to the mother's uterus and can exist for 9 months, or in elephants for 2 years? How can you have something that's not genetically identical to you in you for that long without any immune consequences, right? Well, there are some immune consequences. So typically, the mother's immune system is downregulated, and she can tolerate at least the fetus for at least 9 months, okay? And this is a screenshot from the movie Shaun of the Dead. How many of you have seen Shaun of the Dead? Oh, a lot of them. So for these next, not everyone rose their hand, so it's a zombie movie, and zombies attack, and they have this brilliant plan that they're just gonna go to the local pub and wait for it to all blow over. They're gonna go to the Winchester and then wait for all the zombies to die. They're gonna have a pint, and then everything's gonna be fine when they come out, all right? And so you can think of the maternal immune system like that, like it's not a long-term solution, but we're just gonna deal with it and hope for the best, right? And so there's a downregulation of the mother's immune system and a regulatory response of both the placenta and the fetal cells. But what's really interesting is she can't tolerate it for that long. Soon after delivery, her immune system activates and starts clearing these cells, right? So she's really just tolerating them for a short term. And very similar into the movie, their plan backfires and they have to fight, right? They end up having to fight the zombies. They can't go to the pub and have a pint and let it all blow over. They have to go out and actively fight the zombies. And so what we see in the literature, and they've studied this and they're measuring days after conception, you see this peak. So this is a mouse model right here. So you see this peak and increasing number of fetal cells in the liver, the lung, and the spleen in these mice. And then what's really amazing about mice is they give birth on day 19.5 consistently. How amazing is that, that you can predict? Like, oh, I'm going to have my baby Tuesday, maybe Tuesday night, instead of this like 5-week window that humans have. So mouse models are much more consistent to study. So you see this day 20, this dramatic decline in detection of the fetal cells in the mouse, okay? But that's not the whole story. The immune system isn't that great at clearing all the cells. And you do see some cells that don't get cleared. And we don't know why or how. But the immune system is not completely effective at clearing all the fetal cells from the maternal body. And there's been some really amazing studies to show that these cells are not just floating around confused, maybe the little zombies of their self, right? They go into specialized tissue. Oh no, the blood cell image. Okay, it looks like this blobby mess, only a different color. But you know, they're pretty, but it's hard to interpret. So they're just lighting up the Y chromosome and the X chromosome in these studies. But what they're really showing is that they can specialize into individual specialized cells within the target tissue that they end up in. So they're performing a function. They actually do something in the tissue that they integrate into and differentiate into, okay? And there was a really nice study that came out that showed that microchimerism is detected in the maternal brain. I mean, this is kind of mind-blowing to think about, that you could have cells from your baby migrating to your brain and differentiating into neurons, okay? And they found it in quite a high frequency. And remember, they're just looking at male microchimerism right here. And they're looking at just different regions. Right? So this could really be an underestimate. You're like taking random samples and looking and finding it in at least 63% of the women, they found male microchimerism in the brain. And so this, again, that's the glowing Y chromosome right there, showing that it's a male cell in the brain. The only caveat here I would like to add is this was not sequenced, okay? They know this woman gave birth to a son, and now they're seeing that she has male cells, male DNA, lighting up in her brain. And the assumption is it's from her baby, because we know that there's this transfer of cells. This hasn't been sequenced, and we have no empirical test right now to confirm that this is really from her baby. Yeah, go ahead.
Can I ask a question?
Absolutely.
Do they do this for women that has never given birth?
Yes. Oh, we were going to say that's a question, but I'll answer it right now. So they looked at women who had never given birth to a son. And they found it. Yep, they found it. So first study looked at women who never gave birth to a son and still found male DNA in about half of them, right? But like I said, miscarriages could happen and you never knew, right? And so there could be male fetus. The other thing that could happen is those women could have been twin in utero. The third option, this is kind of, I'll wait. Let's just think about it. There's one more, and I'll get back to it at the very end of the talk, okay? But
Suspense is killing me.
I know.
Having twin in utero seems like a pretty rare event compared to the 50% observation.
Right, yeah. So yeah, I'll get back to that. Okay, I promise. All right. So what are they doing? So we see that they're integrating into these cells. They're just not floating around. They integrate, they differentiate, they become the specialized function of the tissue that they're in. And we also see that, at least in the mouse studies that have been performed, is that they can fluoresce these fetal cells in a mouse model. And then they do some kind of study to injure the ear on the mouse, and they actually watch these fetal cells track to the injury and help perform repair, tissue repair in the mouse model. This is the mouse model. They've also looked at women and took biopsies of their C-section scars and showed that the fetal cells that were in the scar tissue were expressing important things like collagen that were important for repair of the tissue, okay?
Kids are useful.
Wait. But there's a cost and benefit to everything, all right? So what's going on here? Are they helpful, or can they be harmful? So they've also been found in the blood and tissue of women with a lot of different types of diseases. Mostly you can break it down into autoimmune diseases and cancer, okay? For those of you that don't know, women get autoimmune diseases much more frequently than men. And so this was kind of a really interesting component to the autoimmune community, thinking that potentially it could be this microchimerism, or having a baby that influenced the disease onset or something like that, right? But they've looked at samples of diseased tissue and also tumor samples and found fetal cells actively in either diseased tissue or the tumor. And so the big question is, I mean, the field's actually quite confused at this point, and we really don't know, like, spoiler alert right now, we don't know what they're doing. We have no idea. It's very difficult. There's a lot of organs in the body. It's very hard to find. It's a needle in the haystack. But maybe we can use some evolutionary theory to figure out what actually the function fetal cells are performing, what they're actually doing in the maternal body, if they're doing anything at all. I'm obviously making it very provocative that they could be doing something, but a big counter argument is that they're just there and coexisting, and there's no function, and the quantity doesn't matter, and what they are expressing does not matter. Okay, so there is a bystander hypothesis out there, but as an evolutionary biologist, and knowing that placental mammals evolved 100 million years ago, wouldn't it be neat if they actually evolved some kind of functional adaptation to the mother's body, okay? And so you can ask this question, are they helpful or are they harmful? It seems like they're involved in tissue repair, but also they're in these diseased tissue sites, so they could be creating this immune response or aggravating the tissue. Okay. So that brings me to rule number 2 of becoming a mommy, is the babies want your resources now outside of the room. I'm going to transition to what we think could potentially be going on in fetal cells after the mother has given birth, all right? So they're increasing in frequency, the mother's immune system is okay with it, they're having a pint, and then birth happens, mother's immune system decides to clear the fetal cells, not all of them get cleared. What's left? What was selected for, okay? And I know there's a lot of mothers in the room, or parents in general. We know that human babies are very needy, and they need a lot of parental investment, all right? Especially human babies, okay? This is actually a really cool book. It's called Zombies in Love, and then this is a sequel, Zombies in Love 2+1, and it's a great children's book, so highly recommended. Okay. So, human babies are very needy, and we have this prediction that fetal cells are involved in resource transfers to the baby after it is born, okay? There's functions in the maternal body after the baby is born. So if you think about it, how do mothers transfer resources to their baby after they've given birth, okay? One way is through lactation. I know there's going to be a talk tomorrow, or is it later today, on bones and I think it's a nutritional talk in a few days, so. Oh, it is this afternoon. Great. So babies need mother's milk to survive. And so it would be a pretty keen prediction that fetal cells that integrate into the mother's breast tissue are expressing really important hormones for lactation, right? Also, babies need heat to survive. They're very poor thermoregulators. We see a lot of the studies of autoimmune disease in women within the thyroid. And the prediction is that fetal cells could migrate to the thyroid and basically ramp up the maternal, or at least manipulate the maternal body regulation. And then lastly, the brain. We know that human babies need extreme parental investment. It would be something we would predict is that maybe the fetal cells are either migrating to the brain or just hijacking the maternal hormonal network so that you express all those lovely oxytocin hormones that you do express when you see your baby, okay? That's a really good feedback mechanism to make sure that you give investment to that child, okay? And so it leads us to ask, what if fetal cells were the baby's insurance policy after birth, right? Making sure that the mother not only transferred food to it after it was born, but also paid attention to it and kept it warm, okay? It's leaving behind something that can manipulate or potentially regulate this resource transfer and heat regulation, okay? So those are evolutionary predictions, but just building on it slightly towards the end of this, we know that when it comes to resources, like I explained in the placenta biology, you might see conflict, okay? Just as you see the fetal trophoblast cells invading into the maternal endometrium, do we predict at these sites of resource transfer, that you're also going to see that maternal fetal tug of war, okay? And so these are our predictions, that if fetal cells are doing harm, it's going to be in these tissues where we see this maternal fetal escalation of conflict happening, okay? And so within the breast, the thyroid, or the brain, we might see that fetal cells are not as helpful as in other tissues. And maybe putting it in this framework can help tease apart how conflicting the current study of are fetal cells helpful or harmful. It's really that they're both, and it's tissue specific, and it's maternal specific, and it's paternal specific on whether that fetal cell is ramping up beyond the maternal optima and how that maternal responds to it, okay? So that leads me to the third rule, which is the same. Babies want your resources probably forever, okay? And so how to become a mom bee, just remember, they want to suck the life out of you. If you give it to them, they will take more of it, okay? And then I just wanted to come back to some chat that we had earlier. I gave you a very one-sided overview of microchimerism today. I just talked about fetal microchimerism. I told you that there's a bidirectional transfer. There's also maternal microchimerism. There's maternal cells that are being transferred to the baby, potentially manipulating the baby as well, maybe to cry less or something like that. Wouldn't that be nice? But also, there's been some really great work to show that there's this transfer of immune cells. There's this transfer of antigens that help the baby's immune system develop. So there's this bidirectional transfer, swapping of cells. But as you see here, we also have a sibling in the picture, right? We don't know how far this goes. If you start building a family tree of microchimerism, if this baby's cells are still floating in the mom when she's pregnant with the next one, and there's a bidirectional transfer, there is a potential that the older offspring cells could get transferred to the baby. So going back to your question, what are the three different ways, right? So it could have had a miscarriage, the women that never gave birth to a son. Could have been twinning. And the third option is that they could have just had an older brother. Or the mom could have had an older brother. Or the grandma could have had an older brother. Because you see how this could go back forever, right? And so building on that study, a recent study just came out looking at young girls before they went into puberty, all right? So they looked at them, and so they could have never been pregnant, right? And so they could have never had a miscarriage. They still found male DNA in these girls, right? A few of them had older brothers, but not all of them. So it's very complicated, and we don't, until we start sequencing these cells, we have no idea where they're coming from. But we can only assume it could be multi-generational effects, or again, twinning in utero, okay? All right, so I hope I've been able to convince you that you've been manipulated, because, you know, not everyone has been a pregnant mother, but everyone's been a fetus. And so we're all recipients of microchimerism, and these cells potentially could have different genetic interests than yours, okay? And I briefly touched on it, and there could be potentially completely different genetic interests of the different cells within your body as well. And you can go on forever, and it's a really amazing, mind-blowing thing to think about. And it's actually just been, this paper was out in 2017, and the authors, recognizing that there could be this multi-generational transfer of cells, they're now coining the term microchiome, and you can think about it a lot like the microbiome, where there's probably cost and benefits to having a microchimerome, all right? And yeah. With that, again, I wanted to acknowledge the co-authors here. I'm more than happy. I hope to end early enough that I have plenty of time for questions. Just know that I'm 5.2 seconds at being able to amputate a limb. So, more than happy to take any questions. With the hacksaw. With the hacksaw.